And okay. And as Oliver said, today I'm going to present some research that was funded by the Dirkonney Syndrome International Association to support the development of a personalized therapy for a Dirkonney patient with a novel splicing mutation. So before I start, I just want to give a bit of background into n-Lorem for those of you who might not know who we are. So n-Lorem's mission is to apply the efficiency, versatility, and specificity of antisense technology to charitably provide experimental ASO medicines to treat patients with nanorare diseases. So by that I mean less than 30 patients worldwide with the same mutation. So as we know, Dirkonney is caused by about hundreds and hundreds of mutations. But the patient I'm going to talk about today is truly an N of one because it's the only patient that we've identified with this specific mutation. So initially what started as a small focused mission, we're aiming to change the treatments of rare disease and how we think about human health. So for those of you wondering whether your loved one may have a path to an individualized treatment through n-Lorem, I'm just popping up this slide to give you a holistic view of the treatment plan through application through to treatment. So I'll put up an email address at the end of the talk for individuals that would like to speak to their physician to see if your loved one may be amenable to our ASO technology. So what would happen is obviously you'd consult your physician, they would apply to n-Lorem through our website, and that'll go to an access to treatment committee, and if accepted, it will go through our typical discovery pipeline, which involves the in vitro or the cellular discovery, then through to manufacturing, engagement with the FDA, engagement then with treatment and research centers where treatment and evaluation will happen, and then the ongoing treatment obviously will collect data and have detailed aggregate analysis. So like I said, a lot of the research that I'm going to present today was funded by the Dirkonney International Association. So I think Helen just touched on this, so I'll give a bit of background because obviously this is a mixed audience. So the Dirkonney gene and its associated protein is known as a kinase. So what this does is, as Helen says, it functions by activating or inactivating other proteins by adding a specific chemical group to it. And these proteins typically have widespread function and are critical for the normal working of the cell or the tissue that it's involved in. And this is particularly true of Dirkonney because it's expressed in all our cells of our body. So Dirkonney is involved in a key one would be cellular differentiation. So that's the moving from an immature cell to a differentiated cell. One of the big things that I've found involved within this, especially in fibroblasts, is cell cycle progression. It's involved in DNA repair and splicing, and I'll touch on splicing because obviously this is a splicing mutation, so I'll get into that. But for the families in the room, I just wanted to kind of present the flow of genetic information in a manner which hopefully people can understand and then follow through the talk. So we have DNA. Most people know what DNA is, either through PCR testing through COVID or through shows. So if you think of the DNA as the hardware of a computer, you're born with it. It's sort of the thing that comes with the computer. You can't really change it. And then a process known as transcription happens where it creates a single-stranded molecule called RNA. And you can think about this as the software of your computer. So when your computer's functioning properly, you'll always get these updates. Much like the RNA in your cell, it's always turned over. It can constantly be changed depending on the context. And then there's another process called translation, and this produces the protein, so the actual DYRK1A protein. And you can think of that as the document. So if the software in this case is PowerPoint or Microsoft Office, the document will be this PowerPoint presentation. And obviously, my favorite molecule in the world, since I work in RNA therapeutics, is RNA, and it's n-Lorem's focus as well. But there is one more critical step before you get from the RNA into the protein. So as I said, you've got this double-stranded molecule DNA, and then that transcription happens, and we have this single-stranded molecule called RNA. And RNA is, in its immature form, has these regions that are not destined for the final message. Initially, we thought they were like what everybody used to term junk DNA, but now I don't think that that term should be used anymore because there's so much function to them. But what needs to happen is the cell needs to efficiently remove these regions called introns and then ligate or stick together these exons in a precise manner. If they're even out by one base, you're going to have really significant problems. So what they need to do is efficiently do this all the time. And like I said, some RNAs have a turnover of just hours, if not minutes. So they are put into a continuous message. So you can see exons 1, 2, 3, and they'll produce a healthy protein. If a mutation exists, what happens is depending where the mutation is, it can either change where that exon is recognized, and this is called a frame shift. So some of you, if you've had a genetic report and you've got a frame shift mutation, it basically just means that a base is either added or removed, and you've had a change in the reading code. But sometimes what can happen is if the mutation is outside of those regions, you can get a loss of an exon. So you can see here that 2 is now ligated to 4, and then this can cause a diseased protein, or at times cause no protein at all, and it goes through a process called nonsense mediated decay. So now when we're looking at the specific mutation that I'm going to talk to you about in the specific therapy, it's a 1072-1 G2A mutation. So that basically means that it's one base outside of that exon. And this region is really, really critical to the formation of the exon and formation of the message, right? So if you look here at this diagram, AG is the last 2 letters of that word before that line. And the size of the lettering there basically means the proportion of that base in that position. So because the A and the G take up the entire block, it means that 100% of the time you need an A and you need a G. In the instance of this mutation, that G is being mutated to an A, which creates what we call a really weak splice site, or the abolishment of a splice site. And this was shown through some experiments. So I did a lot of work before I joined NLRM, and initially it was thought to create a cryptic splicing event, which I guess you would call a frame shift. But understanding the functional consequences of mutations is absolutely critical in ASO discovery, because if you do not understand the functional consequence of the mutation, you cannot treat the patient properly. So as you can see here, in a control cell and in a neuronal cell, we've only got one band. That means that the message is full, it's complete. But then in this DYRK1A variant, you can see that there are 2 bands, and this correlates to the complete loss of exon 8. So this is just Sanger sequencing. So this is basically just a means by which you can read all the genetic information. And it perfectly correlates to the fact that exon 7 is now ligated to exon 9. But there are things such as atypical splice sites. And as you can see here, this is a typical splice site that is used very, very infrequently. So 0.06% of the time. And even the A on this thing. So if you think about that, that's 0.6 of the time. The size of the A here is the proportion that it is. So it's used even less. But this means that there is hope for this variant that we could potentially get this exon back in. Although we knew moving into it, it might be really inefficient, but potentially even just removing the mutant may have some therapeutic benefit. So how does this look? Again, now I've put the gene, as I said, they're all ligated together. And once the gene message is here, it creates this protein called DYRK1A. And this is the main protein in humans is 754 amino acids. And here you can see that this is the region that we're missing. And as Helen was saying, the addition of those groups is really determined by this kinase domain. And the exon 8 is encoded almost in the middle of the kinase domain. And actually, exon 8 also encodes part of the nuclear localization signal within the kinase domain as well. So this is really what we're trying to do. We're trying to put this region back in to the gene message so that we can put this region back into the protein. So how are we going to do that? So I did my PhD, my postdoc, and my fellowship in ASOs, so I really love them. They're exquisite little molecules. So they're short, synthetic, single-stranded nucleic acid analogs, which are basically meaning to RNA, and they're about 14 to 25 bases in length, and they're designed to be exquisitely selective and specific to their gene target. For those of you who have got some experience in ASOs, and like Helen was saying, they use a morpholino, which is a specific chemistry of ASO. It's what I used to use in my old life. Now we're using what we call phosphorothioate. These molecules, I think most people are aware that they cause gene downregulation. And I'll touch on the ones because the way that you can modify them, you can change them to actually manipulate splicing, or in the case of morpholino, used to manipulate and inhibit translation. So decades of experience with this technology has led to successful treatment of many patients. Spinal muscular atrophy is one of them, which is Spinraza, which is actually used to put an exon back in. And there are other diseases, Duchenne muscular dystrophy. This is actually to remove a disease section. So there are currently patients here that are getting clinical benefit both systemically and neuronally, so through the brain. So in our case of DYRK1A, we really want to increase the gene and protein expression. So like I said, we'll use a fully modified, because the ones that I'm sure a lot of people know, they're called GAPMAs. They actually down-regulate protein, gene and protein expression. So what we kind of do is we mask. Not only does the splice site determine whether something gets put in or out, but there's this process known as alternative splicing, where one gene can make a lot of different proteins just by changing the exons that are put in. And these are generally determined what we call by like splicing factors. And by this, we mean we're going to block the motif. So these splicing factors actually bind onto the RNA and help a molecule known as the spliceosome determine what is going to go in and what is going to go out. And by masking them, you're actually just physically blocking them. So you're actually not allowing them to bind onto the molecule. So the ASO is actually going to what we call sterically block that. So it's actually just a physical blockade of saying, okay, well, I'm bound here, your proteins and your RNA binding proteins, you can't bind here anymore. So I'm going to touch on, obviously this is an N of 1 therapy, but I'm going to touch on the relevance to the community throughout the slide. So the data generated in this study will provide the first evidence that DYRK1A can be therapeutically targeted by ASOs and hopefully increase the gene and protein expression. How much do you say we need? Well, we don't actually know. But in terms of haploinsufficiency, so Oliver mentioned, and for those of you who obviously have loved ones with Dirkonney syndrome, I'm sure you know it's a haploinsufficiency disease. So this basically means that half of the protein, as you can see here, is not enough to maintain healthy functioning. But what happens is there's this sigmoidal curve, so that potentially if we want to see therapeutic rescue, we might not need to get to that 100% because ASOs are never going to be a cure. They're just going to hopefully change from a severe disease to a less severe disease. So, you know, theoretically, 1.2 to 1.5-fold may be therapeutically beneficial. And like Helen was stating, we do really need a biomarker or a clinical assay to be able to determine what we see in vitro in preclinical data and the upregulation that we see of this protein, and especially for a global protein upregulation, this would be very useful because we can say, okay, in cells we got X amount, and then now this has gone into a human and we're seeing this clinical benefit. So I'm just gonna, this is kind of our cellular analysis during our screening, and this is the main proportion of my talk. The validations I'm not going to touch on. This is just to ensure that we can measure the protein and that we're getting accurate readouts. We start with a single dose screen, move to a dose, move to a BJAB, and in the case of the splicing assay, we'll move to a protein study, but I'll touch on these in more detail. So the current study's aims were twofold. We had the validation of the transcript ratio and the protein expression. Because not a lot of literature is out there on a lot of the DYRK1A variants, they're either used in models, but there's actually not a lot of DYRK1A patient cellular recognition of the genotype-phenotype correlation. So we needed to do that first. And then we went into the discovery phase, which is identifying lead ASO sequences to include that exon. So getting into the data, the which to me it wasn't surprising, but to some people it was. The haploinsufficiency actually doesn't appear to be apparent in the gene itself. I can tell you why. So for those of you who are who know RNA, it's because exon 8 is completely in frame. So what happens is once exon 8 is removed, it actually doesn't cause a frame shift. So like I said, changes the way that the gene is read. It's just a portion that's removed. So this means that it's going to escape a mechanism known as nonsense mediated decay. So you can see our Dirkonney patients here. This is the gene expression. And you can see it's actually higher than one of the unaffected that we have, but it's sort of in the middle. So there's actually no difference between the gene expression. Now, this doesn't actually mean that that gene is specifically functional. And as you can see here, then we did a Western blot on DYRK1A, and you can see here that the level of protein in our Dirkonney variant compared to a control is reduced. So the intensity of those bands is directly proportional to the amount of protein that's there. So you can see here that the Dirkonney variant has, we've normalized that to say that that's 1-fold, and then the healthy control has roughly between 1.5 and 1.7 fold increase. So what I wanted to do is I was like, okay, well then obviously the haploinsufficiency is in the protein. So I designed an ASO to remove the exon, so actually cause further disease. And see if that would do anything. And as you can see, it's further reduced the amount of protein compared to uncontrolled. So actually what's happening, and this does happen sometimes, there's a molecule or like it's not really a molecule, it's a conglomeration of proteins called the ribosome that actually makes this protein. And there's data that says like very structured proteins, so like kinases are very, very structured protein. If they're conformation, the way that they're put together is not correct, the ribosome will disengage and not actually translate that protein. And I think that's going on here. But you know, to prove that for publication, we need to do a bit more research. But the relevance to the broader community here is understanding the functional consequences of mutation. I think a question was like, oh, do we have a genotype-phenotype correlation? Well, we don't really. We know it's haploinsufficiency, but potentially what if there's some dominant negative? So that means like, what if a Dirkonney protein, a mutant protein is produced and actually causes the normal protein to not function the way it should be? And could this then relate to severity? Because obviously, as you'll see, like there's a huge spectrum in Dirkonney, so a lot more research needs to be done into that. So now coming more into the therapeutic side, the single dose consists of casting a wide net of ASOs, where we design hundreds of ASOs so that we're very thorough. The single dose is to determine how efficacious the ASOs are. This basically just means how well it can correct the production. So here you can see all these gray, I think they're here in this slide. Yeah, gray lines represent a single ASO. And what we've done is we've micro-walked or tiled the entire intron. So those factors that I said bind onto the RNA here and determine whether this piece of information is included, they generally sit here. So what we want to do is we want to mask that, and you can see that we've designed hundreds of that. So for the brevity of the talk, I didn't want to just show a bunch of graphs that you know, didn't do a lot, because when you do drug screening and you screen hundreds, most of them don't hit, and that's normal. But here is a graph of all the hits that were destined for DRC. So what we want to do is this is normalized to a control that actually nothing's happened, and also normalized to a non-targeting control. So basically an ASO that actually doesn't bind anywhere, but is just there to control for that. So really what we do want to see is a reduction in the red bar and an increase in the blue bar. So that basically means that we're reducing the amount of the disease allele and increasing the amount of the protein. Now you can see there's this region where I'm doing the exact opposite, but this is another internal control that we can say, okay, well, if we can include this exon, let's see if we can skip it. And then these were what we used to determine that there was haploinsufficiency of the protein. So you can see this nice region here that actually corresponds really well with an HNRNP region, which is a major splicing factor that promotes the exclusion of an intron, of an exon. So here by blocking it, then we're masking that. Then we took those leads, those hits, into a dose response curve to determine the potency of the ASO. And basically this just means how much of the ASO do you need to get a strong effect. So the lower the dose, the more potent the ASO is. And this is done in a 5.4-fold manner. So same sort of thing here. What you want to do is you want to see an increase in the blue bars and a decrease in the red bars. And if you've got an exquisite drug that behaves perfectly in pharmacodynamics, you should see a dose response. So you should see, as these bars get more intense, a continued lowering. When we work with ASOs that reduce gene expression, this is very well known, but splicing is very nuanced because there's a lot of factors that go into determining what this is. So the data is generally not as clean as using what we call a GAPMA, but you can see that there is an increase here for these ones, and then these ones are our main leads that we work, where you can see that as you increase dose, you decrease the disease, and then you increase the wild type, and then here are our two controls that you can see that they have no effect. So NLRM has an impeccable safety record to date, and we always want to maintain that because these are experimental drugs. So we know that ASOs can trigger an immune response, and we want to exclude activators of the innate immunity. So what we'll do is we'll typically test 40 to 80 ASOs who have the best therapeutic potential against benchmarks that we are known to cause issues both in cells and in the clinic. And any ASO flagged as inflammatory is discarded from future development, even if it's the best one. If it's the most therapeutically relevant, if it's inflammatory, it's discarded. So again, I'm not going to show you a huge graph, but here we've just got our control, which sits at 100%. Inflammatory, we have some intermediate that we know actually don't cause an issue in humans, but it's just good to know. And any ASO that sort of flags over that red line, like I said, is immediately discarded. But the ones that we took into protein analysis, you can see they cluster really close to the control, which is great to see. So then for the last step, I guess, in our discovery pipeline before we go into animal studies for splicing cases, is to ensure that these genetic modifications that we're doing is actually reflected in the protein. So like I said over and over, splicing is very nuanced. And when I was a postdoc, we would see sometimes the 100% removal of an exon, but that would cause no change in the protein. Can't tell you why sometimes. I mean, that's the beauty of research. It's ongoing. But what we always need to do is check that the gene change is reflected in the protein. So here we've got Western blotting again. Really hate this technique. It's annoying. Everyone does Westerns, they know they can be annoying, and the DYRK1A gene sometimes is unforgiving. So what we can do here is we have GAPTH, which is just a loading control to know that we've loaded the same. We have a DYRK1A control here, and really what you want to do is just see the intensity of these bands increase. You can see for some of them we don't really have much modulation, and that's typical of therapeutic development. When you take a lot of candidates through, you're going to lose some. But you can see, especially on this plot here, that the micro-walked ASOs, so this is really encouraging to see, right? So the ASOs that are in the same region, you see some benefit, and then you continue to see some benefit, and then as the sequences are moved off the specific position, they lose benefit. And the one in red, as I said, is just another control that we like to keep, so showing that if we remove exon 8 from the healthy allele, that it actually reduces the protein even further. And then here's the densitometric analysis, which is basically just analyzing how that is. So we got DYRK1A controls there. And then this UTC control is just an apparently healthy individual. So just to wrap up, we had numerous ones that were found to modulate gene expression in DYRK1A. Not all of them could modulate the protein. We saw a decrease in the disease copy, an increase in the healthy copy. We had to exclude some ASOs from the BJAB, but for the most part, most of the ASOs didn't cause an inflammatory reaction. And as I said, some of the hits caused increase in protein expression, which is great to see. Future work, so this program is currently in tolerability. From that, We generally send about 20 ASOs to tolerability, where these will be tested in rats with an intrathecal injection, so an injection into their spine to make sure that they don't cause any other toxic effects. And then from that, we'll select a lead and a few backups that then go into GLP tox. So this is where you make a really high-grade drug, and then you do a long study to ensure that if you did long-term treatment or dosing, that they're safe. As I mentioned before at the beginning of the talk, if you do have any questions, I do suggest you speak to your physician or your clinician, and they can send an email to info@nlrm.org. And if your clinician or your physician feels like your loved one may be a candidate for treatment, you can reach out there, or they can apply on the website. So obviously, it takes a village to raise a child, as they say, so it wasn't just done by me. This is headed by Medina, and I've highlighted Andrew here because he did a lot of the in vitro screening as a scientist, but everybody else here works on the discovery phase. So this is actually the first initial stage before it goes into those clinical studies. And lastly, obviously, just like to thank our funder, the DYRK1A Syndrome International Association, for the generous award that we got last year. Yeah, I'm happy to take any questions. So thank you, everyone.
Any questions? Thank you very much. It's very good idea. And I've seen this type of ASO also, I think. So it goes out, they're really designed for the human. That's
For the human, yeah.
So the best way to go about
Yeah, so that's yeah, so we're currently doing that because what I wanted to present was obviously what was funded by the syndrome, but we do have iPSCs, we do have neurons. We're looking at how those affect in those cells. So yeah, there's some exciting work that's still going on in the space with these.
And what is also says I've seen that you can identify from those two that I think, but is there any groups with that variants?
Yeah, so they do. We've currently got dozens of patients that are being treated. So the ASOs are administered through an intrathecal spinal tap. So it's not a systemic delivery. So it is injected straight into the CSF, which does allow it obviously to enter the spinal cord, and then it does get into the brain. And even in the mice, they've done a lot of studies in mice where they've used a fluorescent ASO, and you can see it gets really nice into the cortex.
That would be interesting to me. I would show if some very little non-assessed sound for I think that would be a challenge to find compared to the.
Yeah, well, I'm interested. Yeah, well, that's really interesting. You know, like I said, we've got iPSC, so you know, when you present your talk, it'll be great to see which. Yeah.
Hey, Craig, do you mind just repeating the question and just for clarity for someone?
Okay, so the question was just about how the ASOs, initially it was thought that maybe we're going to deliver them systemically and could they cross the blood-brain barrier. But as I mentioned, the ASO is actually administered directly into the spinal cord through a spinal tap, through an intrathecal injection. And then also another thing was that was asked was the distribution and whether we thought we could get nice even distribution throughout the brain.
Hi, it's Michael Winkler. I'm in medical genetics at Baylor College. I wanted to ask, so if assuming all goes well, the idea to then conduct an N-of-1 trial, I guess my main question is sort of what neurological features would be tested in such a trial? Because the reason I'm asking this is this gene in particular, I think the neurodevelopmental role and like treating a patient who's already at a certain age, like if you can get some very important information for the whole community by seeing whether you can have a therapeutic effect, like at a certain time.
Yeah, so the question was, is this going to be administered in an N of 1 manner? And sort of how are we going to measure, I'm assuming, so I'm just paraphrasing, the therapeutic outcome and the data that we would get from here, how it apply to the broader community in terms of what age we might need to start dosing to get this therapeutic benefit. So I'm more on the I'm more an ASO expert, but our typical development pipeline, yes, is an N of 1. So N Lorem is a foundation that provides experimental ASOs free for life for nanorare patients. So our goal is every patient that's submitted, develop a drug for them, treat them with that drug in N of 1 nature. The IND submission through the FDA goes through a special process whereby we will capture. So once we have a lead drug and it's passed all the tox, the patient will come in and they'll capture all of their phenotypic data through either the natural history that their submitting physician has, and then once administration starts happening through a process, we'll have obviously outcome goals. So it might be, in the case here, if the patient has gut issues, it might be like, okay, has it improved their gut mobility? Has it improved dialect? Has it improved autism spectrum disorder? Have you improved the intellectual cognition? That's why I think this is a really important study, although, like I said, it's only N of 1. If we can show the therapeutic benefit, like I said, it'll open up the door to understanding when you need to treat. I know that there's really exciting ASO mechanisms for Angelman syndrome, and they're sort of, I think that ranges somewhere anywhere from like as young as 6 or 7 up to teenagers, and they've seen clinical benefit throughout that spectrum. So although it's a neurodevelopmental disease, I think that the brain is a lot more plastic than we think it is. I think that you can really get benefit from a wider range. Maybe not if you, but actually, no, I wouldn't say that. I don't know, but yeah, there'll be measures in that manner.
Yeah, I think if you look across other neurodevelopmental disorders, it's fairly common that like the earlier they can treat, the better sort of the outcome. But research says that there's still benefit even farther out. And I think one of the about what Craig's doing is this is really a proof of concept to increase Dirkonney protein expression. And I think that problem is sort of groundbreaking. That's sort of what's needed.
Can you give examples of the program?
So can you give examples?
Can you give examples of the program and others?
Yeah, so the question was, could I give examples of improvement in other syndromes? So there's currently like an Angelman syndrome trial that was being run. It had to get stopped because there was a small adverse event, but it's picked up again. And I think from the initial preclinical data, they had some nonverbal kids that then began to not obviously speak sentences, but then improved verbal communication. It improves spasticity, so the irregular movement, and I think some of the cognitive markers were improving, but that's still in clinical trial at the moment. Speaking to other diseases that we treat at NLRM and the ones that are actually in there, a lot of them are epilepsy based. So, we've gone from patients who are having, dozens and dozens and dozens of seizures a day and then dropping them to single. We've also had patients where they had severe ataxia or spasticity in their muscles that were actually able to walk without assistance. So, there is a patient colloquium that's going to be on for NLRM in Boston in October. If you go to our website, if you just look up the NLRM Foundation, I think it will be online as well, and you can see more of the clinical side, because I'm not really in the clinical side. Although I've seen some talks, I'm basically more in the early development stage.
What specific to a decrease in, they say if we let down, they have an overexpression of DYRK1A and it's associated with, you know, seen in an under, or would that be you or something?
Yeah, okay, so the question was what sort of have I seen in the underexpression? Because obviously in Down syndrome there's the triplication. So are you talking about in terms of in the cell?
Like, you know, a lot of research how to increase the expression of
Mm-hmm.
what.
So I think the main thing would be the neuronal aspect. So this is going to be used to treat intrathecally, so to treat the neurological symptoms. I think obviously if it goes through, it can maybe help with the gut as well. But from most of the research, Dirkonney is involved in cell cycle progression and differentiation, right? So a cell will go through a cycle where it'll replicate, produce its own DNA, and then split. What Dirkonney does in the developing brain is it should stop the cycling of replicating cells, and they should then go into a differentiation stage. So Dirkonney is involved in like sort of the G1 S phase progression, which basically means that it's going to actually replicate. So when you increase Dirkonney, at least in the fibers that we've seen in the function, it changes the cell cycle progression. In neurons, I think that they fail to then make mature neurons and then they die. What we hope to do then is by increasing the protein then is when more neurogenesis is happening, obviously the younger you treat, the better that's going to be, because more neurogenesis goes on in earlier on in life. What you would hope to do is by switching and increasing the Dirkonney protein would be make a neuron instead of the fact that it's going to go through this replicating cycle, and you have what they call NPCs, or neural progenitor cells, which can still replicate. What they should do is then exit the cell cycle forever and then just become a mature neuron. Dirkonney is involved in that exit. So what happens is yeah, like the lack of Dirkonney causes less exiting and less maturation. And I think that's also probably why we see microcephaly in the brain, which is the reduced brain cells. In Alzheimer's, obviously, it's overexpressed on a chromosome, and then, as someone was saying, it phosphorylates other proteins. That's why they think it causes Alzheimer's, because you know APP and taus are also on chromosome 21. Is tau on? Is APP on 21? Yes. Tau's not here. No. Yeah, so APP is on.
Katie's asking a clarifying question. Would this therapy be indicated for a frame shift deletion?
No. So the ASO that we're designing, at least these ones, would be specifically only for a patient who had exon 8 skipping. ASOs are very tailored unless they're going through a specific mechanism. So some diseases cause toxic gain of function, and just by reducing that amount of protein, you can have therapeutic benefit. In the case of Dirkonney, obviously, it's the lack of the gene that's causing the disease. So in this instance, because this specific mutation causes this specific consequence, the ASOs that I designed, or not ISO, that we designed, are specifically used to increase that exon. Most other mutations are not going to be a minus. The current patient is the only person in the world that I know has that specific mutation. So unless they have a mutation somewhere there that would cause exon 8 to be skipped as well, this ASO that will be developed is primarily only for this patient.
I think we'll also have a little bit of an opportunity at the end of the day to some questions. We all have some that are written down or something from So we're a little bit behind right now, but that's okay.